spits that out. It grabs a new A, it grabs a new B, it brings them together, it lets go. It grabs a new A, it grabs a new B, it puts them together, it spits it out. It grabs a new A, grabs a new B, spits them out. It goes again and again and again. That's why enzymes make reactions happen so quickly. They lower the amount of energy it takes to happen because they bring things together perfectly, and because they're like a machine, they just keep going and going and going. Guess what? This was not some made-up, oh, cutesy drawing. That's actually a real enzyme. That's a sloppy drawing of it. Here's a better one. It is the one that causes water plus carbon dioxide to make carbonic acid. I think we probably talked about that before with the blood buffering system. That is the enzyme that makes the blood buffer. So they compared it to a lock and a key. This is a big deal. I've got my car key here. Would you agree it's not going to unlock the door, right? Because it doesn't fit. Well, in the same way, lock and a key, very specific shape. The key fits the lock. The substrate fits the enzyme. Most enzymes can only work on one or two substrates. They can only work on things that have, usually one. They work on one thing that has a very specific shape that fits their active site. Good? Make sense? All right. The substrates, the thing that the enzyme works on, the substrates bind to the active site, and the enzyme is not changed by the reaction. It's not used up or messed up. It can keep going and going and going. We talked about that before. You agree, right? Now, I can think of two ways to mess up that door lock. One involves the super glue over in the lab manager's office. Would you agree that if I plug that lock up with super glue, the key doesn't work real well? Okay. You can plug up the active site of an enzyme. We would call a molecule that does that a competitive inhibitor because it's competing for the active site. Are you with me? Yep. There's another way to mess it up. At home, I have a little oxyacetylene rig with the burglar bottles, you know, the small bottles. I could just melt the face of the lock. Now, I might char the wooden door a little bit in the process, but you would agree I could melt the face of the lock. Would the key fit, even if it's the right key? Would it fit anymore after I change the shape of the hole that the lock goes in? No. In the same way, there is another form of inhibition, and yes, we have a slide to make it clear. There is another form of inhibition called allosteric inhibition, where the inhibitor, instead of competing for the active site, it binds somewhere else on the enzyme and causes the enzyme to change shape so it doesn't work anymore. So, also, anything that changes the shape of the enzyme, there's this D word that you didn't see. Anything that changes the shape of an enzyme, what's the D word for changing the shape? Yeah, be loud.
Denaturing.
Exactly, denaturing. If you change the shape of it from its natural shape, that's what denature means, it's no longer its natural shape. If you change the shape, it doesn't work anymore. The 3D shape is what matters for an enzyme. It's what matters for most proteins. So, if you get it too hot, you remember when we talked about the eggs frying and you vibrate the molecule until the hydrogen bonds break and then it unwinds? If you heat it up too much and vibrate the molecule and break the bonds, you change the 3D shape. It doesn't work anymore. You put it into too high of an acid, you mess up the hydrogen bonds because acid is lots of hydrogen. You put it into too basic of conditions, you break hydrogen bonds for the opposite reason. There's not enough hydrogen, and there's lots of OHs, and what happens, the hydrogens that would be used in a hydrogen bond, they'll pick up the OHs and use them. All right, so the shape matters. If you have high heat, if you have high acidity, if you have high base, you can mess up an enzyme. You can change it from its natural shape. It doesn't work anymore. Would we agree that we've talked about the fact that enzymes are proteins? Yeah. We've talked about the fact they speed up reactions often by 1,000,000 times. I mean, much faster. Okay. Would you agree we've talked about the fact that they're very specific? They can only work on one substrate, usually occasionally two. Would you agree that we talked about the fact that they're not changed by reactions, right? Would you agree we talked about the fact they can be used over and over and over again, which is kind of like saying that they're unchanged? Yes, Carter.
Do enzymes have like a lifespan?
Yes, just like any mechanical thing, you can wear them out.
Like, what's their average lifespan?
I think that would vary from enzyme to enzyme. I can't. The reason I'm saying I think is I can't give you a straight answer. Okay. Because it's so not my thing. I'm a dolphin bone guy. So. All right. Are they sensitive to temperature? We talked about that before. Oh yes, most certainly. Heat them up, you can stop them from working. They have a very narrow chemical range as well. Yeah, and what is too acid or too base depends on the individual enzyme. Like the enzymes in your mouth, in your saliva, that start to break down starch when you put it in your mouth, they work really well at that pH. When you get to the stomach, they're disabled by the stomach acid. But your stomach enzymes, things like trypsin that break down proteins, they work really well at that pH, and they wouldn't work at the low pH of the mouth. Does that make sense? So every enzyme has its own specific range of pHs where it works well, its own specific range of temperatures where it works well. Good? Good? All right. They may need special cofactors. We're going to talk about that. But basically, that's where things like vitamins and minerals come in that you need. The enzyme may not take on its proper shape to work until it has a vitamin present or it has a mineral present. Good? They can be inhibited. We talked about competitive inhibitors. There's another type of inhibitor. It's an A word that changes the shape of the enzyme. What is it?
Allosteric.
Allosteric inhibitor. And they're usually tightly regulated within the cell. Yeah, great. Moving on. We've gone through that. Here we have competitive and allosteric. Say the enzyme is supposed to grab onto this substrate and break it apart. Whoa, it doesn't. Not surprising, that's what it's supposed to do. But say you have another molecule that's similar enough in shape that it can get into that active site and plug it up and stop the substrate from getting there. Does the enzyme break the substrate anymore? No. You see why, right? It's just you plugged up the spot, right? Now here they drew it as a perfect fit. It doesn't have to be a perfect fit. If you look at this, if you had something that fit this into the active site, you would shut down the enzyme, even if it left that end open, because there's no way to get the whole substrate molecule in. So this is a competitive inhibitor. An allosteric one works a little differently. Here is the normal process. If you bind to the other site, you can change the shape. If you change the shape, it doesn't work anymore. Would you agree? Yeah, you change the shape of the active site. So macronutrients are big things that you need to eat. Lots of big molecules that you eat in large amounts. The macro refers to the amount that you need to maintain your health. These are things like carbs, like proteins, like lipids. All right. Nucleic acids are not on there for a reason. They have almost no energy source. All right. So. Oh, which I guess I kind of made clear right here. So carbohydrates. Why do you think carbohydrates are in red and the font is a teeny bit bigger?
It's important.
Well, not just that. You are correct. You should know it. But what I want you to know is that carbs are the main energy source in the body. Carbs are the main thing, and especially the brain only runs on sugars, on glucose. The brain needs glucose. All right. You can burn fats or proteins as an energy source. The heart actually runs really well on fatty acids. The heart at rest prefers fatty acids to carbs. Nucleic acids, you need them for building blocks, but they don't have any good sources, and they're not a good source of energy. They don't have anything useful in terms of energy. So a lot of foods have all three of those. Animal products tend to have more protein per gram than carbs. Plants tend to have more carbs than protein. Straightforward. All right. They provide our cells with building blocks. If we take a carbohydrate, we break it down into sugars. If we take a protein, we break it down to amino acids. If we take fats, we break them down to fatty acids and glycerols. Nucleic acids, we break down nucleotides. But is there much energy in this last one? No. All right. And we can use these for building blocks or energy. All of these are used for building blocks. The top three are also used as energy sources. This whole process we call digestion. Moving on. All right. Oh, wow. This was my attempt. I could have deleted the last slide. Here we go. So you would agree proteins to amino acids, and we can use those in new proteins. Carbs broken down to sugars. We can use this for energy or as part of the plasma membrane. Remember, we have the glycoproteins that are in the membrane. They are part protein, amino acid, and they are part glucose, part carbon. So, and then we have lipids, glycerols. We know that we can use those to make things like phospholipids that we put in the membrane. The reason nucleic acids are not on the list is that we don't think of them as a macronutrient. They're only a little amount of energy. We break them down into nucleotides and use them to make DNA and RNA, but we don't use them as an energy source. Are you good? Makes sense. Are you awake? All right. Why can't we get more excitement about this? All right, so metabolism, micronutrients. Organisms need micronutrients too. These are ones that you need little amounts of for health. Things like minerals like calcium, iron, potassium. If you don't have them, you get problems, okay? Sodium. This is an important one. Everybody's like, oh, low salt diet, low salt diet. It's just because we get too much salt. You need some, though.
That sounds not okay.
Very not okay. What happened, and a lot of universities have had this problem. Hey, it's rush week, and wow, we're going to do hazing, and so that's our chance as upperclassmen to torture the incoming freshmen, and it'll be great. And we're going to make them drink so much vodka they vomit all over the place, and we'll laugh at it. Yeah, great. Well, of course, when that happened, they ended up having kids get alcohol toxicity and die. And so a lot of frats, a lot of schools controlling frats, no alcohol during rush week. No alcohol. So then schools, fraternities would decide, hey, all right, no alcohol. Let's make them drink a gallon of water and watch them vomit, and that'll be funny. They don't realize that if you dilute all of the sodium on the outside of the cell, you mess with the ability to conduct nerve signals, and that means that you mess with the ability of the heart to beat. And then you throw the person into cardiac arrest, and they go to the hospital and might die. In this case, the kid died. Yeah, just from drinking too much water. You need small amounts of sodium, you need small amounts of potassium. You don't need to go and all over your food, but you do need small amounts of these things. Sodium and potassium are what are involved in nerve conduction. They're what are involved in heartbeats. All right. Let's talk about minerals and vitamins. Oh, I should have highlighted vitamins in red, but here. Minerals are cofactors. Vitamins are coenzymes. Both of them are the same basic thing. Both of them activate enzymes. Both of them are needed to help the enzyme take on its 3D shape. We just use different names for it. We call it a cofactor if it's a mineral. We call it a coenzyme if it's a vitamin. So what is a vitamin? It's an organic molecule. What is a mineral? It's an inorganic thing, doesn't have any carbon in it. But you take these, and if you don't have them, the enzymes don't have their fancy 3D shape they need in order to work. If you don't have the 3D shape, the enzyme can't do its job. Micronutrients, here we have a good example. We have an inactive enzyme. We add vitamin B to it, and it sits right there, and suddenly we have the proper active site shape so it can work on the substrate. Without the vitamin B, it doesn't work. That's why you need your vitamins. That's why you need your minerals. They make your enzymes work. Why does a lack of food lead to malnutrition? Well, food has the nutrients, the things that we need, has the energy, the stuff that we need. If you don't get that, you don't have the nutrients that you need to make the enzymes work. You don't have the energy to power the cells. It doesn't work. That's okay. I've been bitten. Ow, he has teeth. Ow, ow, ow.